It's my great pleasure today to introduce Anatoly Buchin from the Allen Institute for Brain Science. Anatoly has been working in computational neuroscience for the last 10 years. He started his life studying physics in St. Petersburg, Russia, at the Polytechnic University. And then he moved to Paris at the École Normale Supérieure, studying computational neuroscience with Boris Gutkin. And then he came actually to the UW to do his postdoc with Adrian Fairhall in the biophysics department. And so now he's been at the Allen Institute for a few years and is working as a scientist. I find his work actually really interesting. It's starting with just single cells and the electrophysiology, but then looking at much larger models, integrative multi-scale models, and actually getting back, to real disease. And also, I think the connections to the genomics are also very fascinating because I'm sure Anatoly will talk more about it. But I think now in this new world, we finally have the tools to, both at the genomic level and at the electrophysiology level, to really start having great diagnostics and models for disease to start getting closer to interventions. So really looking forward to the talk.
Thank you very much for everyone who come, and thank you, Chris, for this great introduction. So I'm very pleased to give a talk here, and I'm looking forward for more potential interactions between Microsoft and Insertion Allen Institute for Brain Science. And to start, I just would like to show this picture. You probably know these people. That on the left is Paul Allen and Bill Gates. And they both founded Microsoft in 1975. And later Paul Allen went out of Microsoft, but he also is involved into research because he funds multiple research projects, like Allen Institute for Artificial Intelligence and Allen Institute for Bioscience in general. So since not everyone knows where the Allen Institute is, I just would like to give you a brief introduction, what we do. And if you have any questions, or at some point my talk will stop making sense, just don't hesitate to ask questions. So Allen Institute, after Paul Allen. So we are located physically close to the Mohai Museum and the Lake Union. So this is physically where we are. This is how the buildings look like. So Allen Institute has different divisions. There are 3 of them that are present for now. Brain Science, Cell Science, and Frontiers Group. So I mostly work in the Allen Institute for Brain Science, where we do different types of neuroscience research. There is also cell science that mostly works on the molecular biology scale. When there recently has been released several products, including single-cell atlas. And there is Allen Frontiers Group that is aiming at funding of various research projects around the world that seems to be promising. So, but I'll be specifically talking about the brain science today, because I work there. So probably this is one of the almost textbook definitions that we have these days, is that the human brain is probably one of the most complex piece of matter in the in the universe. And at the Institute, our mission is to improve understanding of the human brain and its norm and its disease, and share our knowledge with the global community of neurosciences in the whole world, not just the US, not just Europe, but everywhere. So how we do that? So the model of the Institute is approximately the following. So at the Institute, we do research. We generate the data sets that are standardized and labeled. And we develop the software tools to assess these data sets. And then we share these data sets with the global community of neuroscientists. And at one side, and at the other side, we write the research papers based on the patterns that we see in this data. And we create the novel computational models, which I'm specifically going to talk about. And they all become freely available. Like the models that I'm talking about will become available this September, for example. So we have data releases and software releases that come periodically. that we share. Also, it's important to mention that Allen Institute is a non-profit research organization, so everything that we do for the global community of scientists around the world. But how exactly does it work, actually? So we mostly interact with people using our website, brainmap.org, and since 2003, when Allen Institute has been founded, there were multiple projects that has been used. For example, there is a big research team about the cell types that tries to determine the definition of a cell type in the human and the mouse brain. There is a brain observatory that deals with the calcium imaging of and tries to understand the computations that take place in the visual cortex of a mouse when it's watching different images. And there are many more projects that I just don't have capacity to talk about all of them. But specifically, we just recently started the translational direction. When we study human brain, one of the web products is already available on the website. It's Human Brain Gene Expression Atlas, where you can search for a particular gene and see where and what part of the brain is it actually being expressed. And currently we started the new direction that corresponds to human epilepsy. But, yeah.
Quick question. How much data are available for the human brain genome, the RNA?
You mean how?
Like how many tissue samples? And how many...
I would not be able to get that straight on the top of my head. You can check on the website, but there are a lot of them. And additionally, last year we released the data set with the human neurons from cortex. So there are over 100 of neurons recorded. But for genomic data, we have much larger capacity because it's relatively easier to generate at the scale. So for human epilepsy data, this data has not been shared yet, but it will be shared soon, as I have managed. So, but what do I mean by human epilepsy? So how actually does it look? So here are a recording of EEG pattern of this very young, unfortunate epilepsy patient. And at some point, when the seizure starts, it's accompanied by highly synchronous activity in the brain that can be measured in the EEG sensors that are placed in the brain. And then this activity have very usually at the time when seizures take place, it's impossible for this person to do anything. They just suffer, and it's good when they can find a very calm place. The seizures might take place up to 20 seconds or up to minutes, and very often people are just losing their consciousness. And one of the biggest problems of seizures is that they are completely unpredictable in their onset. So basically, these people cannot drive their car. And then when the seizure ends, at some point, the synchronous activity stops and all EEG channels, and then the person is just slowly recovering after that. So, but what are seizures and what do we mean by them? So, biologically speaking, this is like a very large umbrella term. This is a group of neurological diseases that are characterized, but all of these diseases are characterized by seizures. And seizures, in the brief version, are the episode of symptoms due to the very highly synchronous activity in the brain that might start in one part of the brain and then propagate to the other parts of the brain. And in fact, it affects a lot of people worldwide, around 50 millions. And in the US, about 12 million people are affected by seizures. And in the adult patients, about 50% of epileptic patients have temporal lobe epilepsy. So this epilepsy that often originates in the hippocampus, and then it spreads through the cortex at different parts of the brain, up to the motor cortex, where people might start having convulsions. The problem with seizure research, there are many of them that about 40% of the patients do not respond to the pharmacological treatment. So they come to surgery only at the time when you cannot treat them pharmacologically. And this is the tissue that we are studying. But the natural question that we might ask is why we cannot pick up the good drugs. So this is the picture from the research paper, which I like a lot, that shows the number of drugs that has been generated over more than 100 years of history, of scientific history, starting with bromide and pentobarbital up to the all modern drugs. So as you can see, we constantly generating more drugs against epilepsy. Yet very often they, up to the 30 to 40% cases, both in the 70s and 2000s, they doesn't seem to work. Why? We don't know. We are just trying to understand it, but it might be related to the case that a lot of seizure models that people are using are tested in animals. So this is like a workhorse of the biotech research, that people use mouse epilepsy as a proxy for human epilepsy. And this approach has been used in many research studies, but for epilepsy, it constantly fails. Another problem with the drugs being used against epilepsy is that they're usually very dirty drugs. They are not specific to this particular region, therefore they have a very strong side effects. So this...
What determines those generations? What are those different generations?
If I'm not mistaken, different companies were starting generating different groups of drugs. Yeah. And basically now we might have some more drugs, but this percentage of 30 to 40 patients of epilepsy, patients who don't respond to pharmacological treatment is relatively constant. So this motivates us to study the human brain itself, because it might be that human and mouse brain share a lot of similarities, but there might be some very substantial differences that don't allow the same drugs affect and the same drugs be applied for the human brain. So the tissue that we get at the Allen Institute comes from Swedish Medical Center. So this is the tissue that we get from pharmacoresistant epilepsy. So these patients have been tried different drugs current of in the state of the art, but many of them, well, all of them didn't work, to be precise. So they decided to go under the surgical treatment, where the whole part of the brain region is being removed. Usually, seizures, as I mentioned, in about 50% cases, are originating from hippocampus in the adult patients. Usually, this part of the brain is responsible for formation of the new memories, but in the pathological case, it might generate epileptic activity. And there are many pathophysiological processes that take place in the tissue. So in this research project, we did aim to describe one particular dimension of this. So we look into so-called sclerosis. So usually when people say that the tissue, hippocampal tissue is sclerotic, this means that the single cells start to disappear from this tissue because of various reasons, including exotoxicity. For example, here is the mild stage of sclerosis, characterized by so-called Weiler grade. When you have a lot of cells, this is the piece of the tissue that has been stained with antibodies specific for the neuronal bodies. So we can see that there are a lot of cells, but as we go into the more later cases of sclerosis, the cells start to disappear, and they're being replaced with the glial tissue. The question that we ask, so apart from disappearing of single cells, what happens with the single cell electrophysiological properties, morphological properties, when we know that this region of the brain is definitely epileptic? Because once they remove it, this patient stops having any seizures. So to answer to these questions, we apply a set of tools that has been developed at our institute. So this is mostly electrophysiological tools, and so we do the electrophysiological analysis and morphological analysis. And I know that in this audience, not everyone might be familiar with neuroscience, but I'll try to briefly describe what it means. So basically, when we have slice of the human brain, of this particular slice of the human hippocampus that comes into the dish, where it's when it's located with the artificial cerebrospinal fluid, so we can fix this tissue and record the electrical activity of single cells. So we do this by the technique called patch clamp, when you put a very tiny glass electrode and attach it to the cell, and then you can inject the current into the cell. For example, you can inject the step pulses, like negative and positive. When you inject the negative step pulses, the membrane is being hyperpolarized when it goes down. And when you inject the positive step currents, it becomes depolarized and it goes higher, up to the level when the single neuron can generate the spikes. Once this part has been done, we inject the special marker that is called biocytin into the cell that spreads through the whole dendritic tree. And then we can use this image stack from the slice, and the imaging team takes it takes it, makes the images, and then we do the reconstruction of single neurons, both manually and using the algorithm as much as it's possible. And then we can resolve the spatial structure of this neuron in the slice. So these are two main data modalities that we deal with, electrophysiology plus morphology. Yeah.
How many neurons are we talking about in this tissue?
So it's not an easy answer, but it's a very logical question to ask. So several, I would say that in the whole slice there's 10 to 50,000 of them. But we can record from very small subset of them. From each slice we can record for 10, 15. Another interesting thing that this human tissue can live up to 3 days, so you can continuously record from it as soon as it stays alive. So the question that we want to ask, so what happens with the single neuron properties when we come from mild sclerosis to the severe sclerosis? And specifically, we'll be talking about one single cell type that we are probing. This is the hippocampus granule cells. And so what happens when we come from mild to the severe sclerosis with the single cell features, and how can we characterize that? So one way to answer to this question is to first analyze the electrophysiological properties. Are the cells becoming more excitable or not? To do that, we do this analysis the following way. So here are two example cells, like one cell that received the current step that looks like this and responded with spikes that are generated at these times. And there is another current step for another cell that we know is Weiler grade 4. So it's much more severe case of sclerosis. So essentially, this is labeled data. So where we can say that this is mild sclerosis versus severe, and neuropathologists can tell it based on the grading scale that is established in the medical field. On the next step, we take and measure characteristics of these impulses. Action potential amplitude, the threshold when the spike is being generated, action potential width, frequency of the spikes, adaptation index, which is determined as the particular combination of the intervals between the impulses. Overall, we have characterized this 30 different characteristics for each sweep. So, and we do this by using the software that has been developed at the Allen Institute. It's called Allen SDK, that is freely available for everyone who would like to analyze the electrophysiological data. But what it allows you to do, it allows you very quickly to extract these features. Think about them as every sweep is becoming a vector. On the next step, we generate the matrix that corresponds to every particular neuron. So I should say that we don't have super large data set because it's human data. It's very rare, but this is one of the largest with multimodal data modalities that we have. So each row here will correspond to one neuron, 1, 2, 3, etc. And each column here will correspond to the particular feature, like time to the first impulse, the number of impulses, etc. There are about 30 different features that had to be manually developed. And the next step, once we form this matrix, essentially we have the labeled data. We can add additional column that will say what Weiler grade is it, Weiler grade 1 versus Weiler grade 4. And the next step to understand what is happening in terms of electrophysiology with the cells, we apply the standard dimensionality reduction tools for this data set. So again, here are our cells that respond to the current step and have a bunch of feature characteristics that characterize the response. And when we do PCA on this, we can find that there is a very robust clustering. So VG4 cells, which we have 23, are more in this part, and VG1 cells are more in the right part. And the line here corresponds to the kinase clustering in 2D space. So initially we had 30 by 50 matrix, then we can project it to the PCA. Also, when we do the t-SNE analysis, we can find the separation of the electrophysiological features between VG1 and VG4. Some VG1 cells are more closer to VG4, but it's expected. And the big advantage of this is that we have this data labeled, so we can actually test how the characterization with the Weiler grade correspond to the electrophysiological features. To study these features in particular, to find what features are the most distinct to characterize the Weiler grade, we train the random forest classifier the result based on the PCA data. So we take this data from PCA here, put it into the random forest, and then we look into the weights of random forest classifier. And the green weights that are located here are the most distinctive one that allows us to separate between the VG1-1 and VG1-4. Additionally, we did a pairwise comparison. So instead of using the statistical technique, like here, we do the pairwise comparison feature by feature between one group and another. And this is, of course, with correction for multiple comparisons using Bonferroni correction. So each statistical feature is marked with a dot here. And if the feature is the same using pairwise comparisons and random forest, then it's green. So essentially, what this technique allows us to do, and here's the significance level for pairwise comparisons. Like some features are statistically significant, but not very distinctive in terms of random forest classifier. So what these techniques allowed us to do is to find the electrophysiological correlates between viral grade 1 and 4. So basically, we can say that if you will give me a first interspike interval and time to the first spike for VG1 and VG4 cells, I would be able to tell you whether it's early stages of sclerosis versus late stages of sclerosis. But electrophysiological data and excitability is just one part of the story. So on the next step, we went to the analysis of morphological features. So we used the feature extraction software that we had developed at the Institute. So here are examples of a single cells, of granule cells. As you can see, they have very distinct morphologies. Also, they come from different patients. So specifically, we have 2 patients for viral grade 1 and 3 patients for viral grade 4. Oh, sorry, 3 patients for viral grade 1 and 2 patients for viral grade 4. And we extract the features. There are 99 features that we used and applied the same trick. So we applied PCA and TSNE, and just by visual introspection, first we were not able to find any structure in the morphological differences between VG1 and VG4. We trained the random forest classifier. We can actually determine it with the 70% of accuracy, whereas VG1 versus VG4. But when we do the pairwise comparison, none of these features pops out to be statistically significant. So which means that there is likely no statistically significant difference in the morphological structure between the early versus late stages of sclerosis.
Usually they seem different, but what kind of features do you extract?
They seem, that is true that they seem different. So we extracted various of them. So like the number of branches, the length of the branch, the top branch, the apical dendrite, the bottom branch, the basal dendrite, and many other characteristics like the size of the soma. It's true that they are very diverse, and there are just 4 out of 20 data sets, and they're all been sampled in different parts of the dentine gyres. There are some morphological variations of the single cells. But we try to pull everything together because if we're not doing this, we'll not have a big enough data set to analyze.
So viral grade 1, are those normal patients where they just have small symptoms of low symptoms? They have some epilepsy, but or are they normal?
All these patients have certain amount of epilepsy. So basically, otherwise you will not get, this tissue will not be taken out from them. So the lack of these studies, which comes inherently, is that we don't really have a real control. Because to have a real control, we would need to pick up the same piece of the brain from a healthy person, but they will not donate it for us. But our way to look at it, we take the amount of sclerosis as a direction of the disease development and compare early stage of sclerosis versus late stages of sclerosis. Yeah. So basically all of the data that I will be talking about when people already have seizures established.
Do you know what's driving the sclerosis?
We don't know, but I can speculate on that. So presumably there is such a phenomenon that is called excitability. When neurons are being activated too strongly, they start to die. And this is a very known characteristic of the epileptic regions. And when they die, the connections also start to disappear. So it might be related to that, or it might be related to some other processes. I would not speculate more further than that. Yeah. But yet here we found that there are electrophysiological differences, and then we found that there are no morphological, but neurons in the brain, they don't work alone. Like in the human brain, they're about the size of a neurons, the number of neurons as the stars in the Milky Way. This is the same level. So we need to have a lot of neurons. Yet, using our experimental technique, we can look into the very small subset. So neuron is receiving the inputs from its neighbors. And it's receiving this input using synapses. So we were wondering if there is any particular difference between the number of synapses. So basically here we use the NeuroLucida software. This is the work of Rebecca De Frates, who just used the image stack that was generated for extraction of the morphological structure of these neurons. And then she looked into the initial part of the dendrite that is in the top, called apical dendrite, and to the tip of the dendrite, and she counted the spines using this software that allows you to feel the shapes based on certain criteria. And the same has been applied for wider grade 4. And here, when we pull this data together, here we can look at it separately at the apical dendrites and basal dendrites. But when we pull this data together, we can see that wider grade 4 neurons have about twice as many spines as our grade 1 neurons. And that's important because the spines is essentially where this neuron is receiving its excitatory input from the other cells. So, and if the single neuron is receiving twice as much input as another, as wider grade 1, so it might make this single neuron much more excitable, which might represent particular link to the pathology. So, to recap just the data analysis part, so we found that there are electrophysiological differences between VG1 versus VG4. We found that there are no morphological differences between VG1 and VG4 neurons. And what is one of the most interesting parts, that we found that there are spine density differences between VG1 and VG4 cells. So it seems that VG1 and VG4 cells have some statistically significant difference, not in how they look in 3D space, not in terms of their morphology, but in terms of their biophysics that makes this neuron different.
Do the patients progress from 1 to 4, or is it just different patients have different grades?
Patients usually, well, the suggestion it is that patients progress from 1 to 4 because they gradually start losing the cells. Yet the speed of this progression is usually, it's hard to predict. You cannot say that earlier patients, like younger patients, will likely have wider grade 1 versus late patients will have wider grade 4. So it depends from patient to patient, but it seems that there is likely that there is a progression because you cannot lose neurons immediately. And it's also important to mention that the data that I'm talking about, so we took the neurons that we can sample and the dentate gyrus where the cell loss is not so strong. So we can just pick up the cells that would allow us to form conclusions. Yeah.
Are any of the morphological features related to electrophysiological features or vice versa? Do you consider them completely independent, or? Yeah, I'm kind of curious about that.
Yeah, that's a very interesting question, if there is a link between morphological and electrophysiological features. There had been work by Mein and Sinovsky from 1998 that showed that there are these links, and by modifying this morphology and electrophysiology, you might change excitability. There might be some, and we are trying to understand it better. There is a person who worked with Rohan, who tries to use the autoencoders to combine the data modalities like electrophysiology, morphology, and RNA sequencing together to try to bring these things together. But for this project, I'm not able to tell you exactly what is the direct link. But they're part of the same neuron, so there has to be a connection. So, okay. But once we studied this data, it allowed us to generate certain hypotheses about what is wrong with the epileptic tissue. But because this tissue is so scarce, we cannot repeat all potential experiments that we would like to, because it's very rare. So we turned into the computational modeling. But before you start making a computational model of a single neuron, you need to find a good model, and that's not an easy task. So basically, one of the classicals used to model neurons in the computer using biophysically realistic models is to represent the single neuron as a cable. So here is one VG1 granule cells. As you can see, it has very elaborate morphology. So this is the soma, where the action potential is being generated. Here's the dendrite, apical dendrite, which is in the top of a neuron. Here's the basal dendrite, which is more closer in granule cells, that is more here at the bottom of a soma. But both of them in dendrites, they go up. And this is an axon. Axon usually has multiple collaterals, it branches. It's very thin, so we are not representing the whole axons because it's too thin for our imaging techniques, but we definitely represent the whole dendrite. And the way how we can generate these models is first you take the whole reconstruction, you discretize it using small steps, usually 20 micrometers that we used, into the different parts. Then you form the equivalent cable model when you set up the model that corresponds to the superposition of cables of the same radius. And then once you have this cable representation, you use the compartmental model. So instead of a cable, you bound a bunch of microcircuits together that will represent the properties of a cable. So basically, neurons are generating electricity. How they do that? They do this by sodium and potassium channels that are located in these neurons. You can think about the membrane of a neuron as a capacitor because it separates the electrolytes that are inside of a neuron and outside of a neuron. And there are some conductances that become activated once you change the membrane potential that make. I cautiously made it very simple. If you want to understand more about the generation of action potential, I have a lot of things to tell you. But for now, just consider a single neuron as a cable. And that is limited by the morphology that we generated. And we adjust the parameters of channels, or of these parameters, to match the electrophysiological data. So we have morphology at one side, with 3D structure of a neuron, and then electrophysiology, how the neuron responds to electric current. And these computations take a lot of time, so we use genetic algorithms. This is the work by another scientist, Ani Nandi. We work together. So in the beginning, you start with some parameter distribution within a certain range, and then gradually, so this is the experimental recording in black, and this is the fittest individual from the population. And then gradually, after several generations, within 100, we are able to find the single neuron model that can represent our electrophysiological data the most efficient way. As you can see, it takes a lot of time, and usually every computation is about 40,000 core hours, and the supercomputer Cori. So we do the collaboration with NERSC, supercomputer center in Berkeley, and we use the software that was been developed by Werner van Geet in Blue Brain project. It's called BluePyOpt. This is the Python library that allows you to access the genetic algorithm. And then at some point, gradually, we start having a single cell that can properly represent this neuron. And I'm just showing one sweep, but we train it in multiple sweeps that don't have spikes and have spikes. Yeah. So, and then once we generated the single neuron models, the natural question is, what are these computational models for? What can they do for us? Because just having a model that can represent the data is not enough, probably, to make a discovery. So we need to have a particular research case to use these models for something. And to do that, this is the current ongoing work. So the output of a model, if you think about it, is a parameter vector that characterizes the activity of the single cell. And this parameter vector corresponds to the particular conductances, like voltage-dependent potassium channels, calcium channels, and many others that are specific for this particular cell. And this combination of conductances allows us to reproduce the experiment and the model. So we don't know conductance distribution by default, but what we know that it has to be in a certain range in order for neuron to make this function, to transfer the input current into the output spikes that it will send further down the pathway. So what we are currently searching for, we are looking for the differences for particular ion channel combinations that would tell us why do we see the different electrophysiological features in VG1 cells versus VG4 cells, which we can find by dimensionality reduction tools. So, because dimensionality reduction tools in our case just show that they are different, but why they are different? And since neurons are highly nonlinear systems, we can look into the parameter vectors and say exactly. Of course, every channel is not coded by one particular gene. Like, for example, HCN channels has about 4 different ones. But at least we are able to, this parameter vector is relatively small. It's about 60 different parameters per model. While in RNA sequencing data, we might get up to 40,000 different genes. So these models might represent the natural dimensionality reduction. So when we look into the, when we compare VG1 and VG4 cells and see what channels are particularly down-regulated or up-regulated in early cases of sclerosis versus late cases of sclerosis. And we are currently under investigation of this link of these channel densities and genes. So another use of these models is simulation of part of the pathophysiological scenarios. So basically here at the top, we have a single neuron that comes from VG1, early sclerosis, that has a synaptic input that is distributed in the dendritic tree. So we randomly put synapses, and each synapse is activated by the Poisson process. So basically, it's randomly activated, and all the synapses are randomly activated, while the synapse density, how many of them we have to drive the neuron, comes from our spine estimation. And this neuron gets about 114 synaptic inputs that are estimated based on spines. And we also know that VG4 cells have a different morphology, well, have presumably the same type of morphology, but slightly different electrophysiology. So we pick up this cell, distribute the synapses and dendritic tree, and we put more synapses. According to the synaptic density and the length of this dendritic tree, it's about 400 different synapses. And in this case, we can see that this single neuron generates much stronger electrical activity in response to the synaptic stimulation. But is it because of different biophysics, or is it just because the synaptic input is very different? And the models can leverage as the opportunity to answer to this question. So basically, we can take VG4 model and run it with the synaptic stimulation the same as VG1 model. And then we can see that even when this single cell is receiving exactly the same synaptic input, it leads to much higher level of activity that we can see. So which points us to the potentially difference in terms of the biophysics of a single cell and their channel expression between this cell and this cell. So we can test various hypotheses using this approach. But of course, epilepsy and seizures are network phenomena. Single neurons don't work alone. So once we have this, and as we have the models that can represent our data, we can take these models and start generating the networks and see what will happen if we'll start changing the biophysics of single cells together with morphology. To do this, we are using the brain modeling toolkit that has been developed at our institute. This is a very powerful software package that allows the Python interface to put these neurons in the 3D space according to the particular distribution, connect them and simulate them on the cluster or on a local computer. So this is a highly scalable tool that we released open publicly recently. And so we are using this tool in order to generate networks of cells. So basically the network that we want to stimulate, as I mentioned in the beginning, so the seizures come from this area that corresponds to the human hippocampus, which is in Latin hippocampus means seahorse. So because it has seahorse-like shape. So when you cut the seahorse in the middle, then you will have this region. And different parts of the brain have different names. So this is part of the hippocampus. This is subiculum, this is dentate gyrus, CA1, and many others. And this is the picture from our neuroanatomy atlas that has been generated. So we take this picture, and we want to simulate the dentate gyrus. And dentate gyrus, like in the normal conditions, it presumably works as a gate for memories. So it's basically, it receives the input from the cortex and then passes it further in the hippocampal pathway, where the place cells are located. So, but we are mostly interested in the behavior of this small local network of dentine gyros, because about the small local network we know a lot from the rat studies. So, in order to generate this model, so because we know that we cannot simulate the whole brain, there are too many cells. So we started with the small networks that can be scalable to bigger networks. And if you are able to see the phenomenon in small networks, we are able to scale it further. So basically, according to the anatomical estimation, this network consists of 500 granule cells and 6 basket cells. Granule cells are excitatory, and basket cells are inhibitory. And both the cells are receiving the external input from the cortex that activate pyramidal cells and interneurons. And when we assemble this network that follows the realistic connectivity that is known from rat studies, and we assemble this network that consists of only VG1 cells and connected in a particular way. So this is an example of presumably physiological behavior that this network might have. We get the external input that activates the subset of granule cells. There are mostly granule cells in this network. They are all red, and there are some inhibitory cells that are shown in blue. Just 6 of them. But it's important to mention that inhibition works in an interesting manner because inhibition affects the soma of a single cell, while this excitatory inputs comes to the dendrites. So inhibition is much more powerful because it can inhibit action potential generation very rapidly. So external input activates the subpopulation. Then the whole network generates the population spike. And then because of activity of the inhibitory neurons, it just brings everything down. Additionally, neurons are receiving some background synaptic input in order to have some activity, because in the brain there are no silent neurons. They're always continuously generating some spikes that might be not related to the stimuli that they received, but are always in the state when they're ready to transfer information. And this is accompanied by very sharp population spike when they generate a burst of activity. And physiologically, you can think that this network received some input from the cortex and passed it further to the hippocampal pathway. But what will happen, we will start changing the number of cells. So when we come to, say, 50% of VG4 cells, we keep the network structure exactly the same. The only thing that we change the morphology and electrophysiology of single cells. And when it happens, it leads to generation of the spontaneous activity. So basically, this network can generate the population spikes when there is no stimulus being presented. And when it keeps increasing the number of VG4 cells, it leads to the next population spike that is even earlier. So in total, we have seen that when we start increasing the number of VG4 cells, which we think are more excitable, it leads to the development of seizure-like oscillation, of potentially seizure-like oscillations. So basically, putting VG4 cells, because of their electrophysiological property, putting VG4 cells into the VG1 circuit makes the circuit more excitable. And why is it so? Because of their electrophysiological properties that we have discovered using PCA and TSNE analysis. So the conclusions for this part of the talk, that we developed the realistic single neural models of human granule cells for human hippocampus that can match our data and represent the single channels that we studied. And we found that VG4 cells are more excitable than VG1 cells because when they receive the same synaptic input as VG1 cells, they generate much more spikes. And also we found the addition of VG4 cells to VG1 network generates some spontaneous activity, which might likely be related to seizure generation.
So you mentioned inhibitory cells. Are they part of the model?
Yeah. Yeah, they are located without the.
I see. Got it.
Even though there are just 6 of them.
So it's constant between WG1 and 4.
Yeah.
The same number of
Yeah.
inhibitory cells.
You might also think that they are very weak in some sense, but they're affecting the soma, and each of them corresponds to about 140 granule cells that are randomly distributed. And this is enough. So basically, if they get too much activated, then they shut everything down. Yeah. Yeah, so the circuit properties remain exactly the same. The only variable that we change is the biophysics of single cells and their morphology. Yeah, so of course, this work would not be possible without the team that I'm working in. So under the supervision of Kostas Anastasiou and Tom Chartrand, who just recently joined our group. Therefore, he's not in this picture. So this is like applied science team at the Allen Institute, and we are constantly searching for collaborations and better use of our data for research. But of course, we are just part of much larger ecosystem. We are not alone. Here, the institute is intermediate scale. It's between the research lab and the big company. So here we have about 700 people, and everyone is doing some particular type of the job. They're not computational people. Is just a small subset. Mostly, it's experimental institute, but we try to make sense of this data and share it with the whole world. And of course, I would like to thank Paul Allen for his vision and support. And I would be happy to answer to your questions in the resting time. Thank you. Mm-hmm.
So you mentioned the need to potentially move to a human model. Given what you've done so far, have you noticed differences between what you've seen here and the mouse model? Or?
In this, that's a very good question. Thank you for asking that. So the question is whether we can see the difference between the human versus mouse cells. We just started working on this, and the recent paper that we resubmitted already to Neuron deals about the difference of HCN. Well, we looked into the cortical neurons of human, layer 2-3, compared to the mouse neurons, cortex layer 2-3. And we found that human neurons have resonant properties that are much higher. So basically, they have a resonance around the theta frequency in terms of subthreshold oscillations. And we also found that they have much more HCN channel, for example, compared to mouse neurons. And we know that HCN channel is often involved in many epilepsy pathologies. So we started seeing the differences that are significant for the neuron types of the same class. But of course, first you have to determine the same class of neurons to make this comparison. But currently we are working on investigating more differences that might be useful for medical research, for example. Yeah.
Maybe a softball. Where are you in this picture? Do you know?
Yeah, I know.
Oh.
It took me a while. Or I knew. Here.
Oh, okay. Yeah, I see the resemblance.
Yeah, here's Christoph Koch, who is the chief scientist of the institute.
Is Paul Allen here?
No, Paul Allen is unfortunately not here. Paul Allen comes to the scientific advisory council meetings and sometimes appears.
There, is that?
Where?
In the white shirt to the right?
No, this is Alan Jones. This is also Alan. Yeah, he's Alan Jones, who is a director of the institute. Here is the Christoph Koch, who is the chief scientist. Before he was a professor in the Caltech, and then he moved to the institute in 2011 for. Yeah.
Do you have something like calcium imaging of a large population during epilepsy to see how realistic your models are? So you're saying that these seizures might be, your model might be predicting seizures? I'm wondering how.
That's a good point. Yeah. The question is if you have calcium imaging. So if you have calcium imaging recordings and whether we can use it for seizures, we can definitely try. And moreover, there is substantial part of the institute. It's called MindScope. When there's a brain observatory project, when people look into the visual cortex of a mouse, when it's looking at different images, trying to understand what does the mouse see. So this might be repurposed, but the works that I know, for example, in Columbia University, when they do calcium imaging in seizures in living mice, you very quickly get oversaturation of the signal. Because calcium sensors, usually they're created to solve one single spikes when the cortex is in a functional mode. Once you have calcium imaging in the cortex, you can still see seizures. You can see the rapid onset, and then everything is blank. So like seizures versus not seizure. And our work is with human. Of course, we cannot do calcium imaging in human because we still don't have the genetically engineered human. But we might compare it with electrophysiological data.
You also have these sensors, I think they're called neuropixels.
Yep.
Yeah, so how about those? Do they saturate as well?
No, neuropixel probes has been recently introduced, and there is a Nature publication about that. So neuropixel allow you to record simultaneously in parallel for over several hundreds of single neurons. You can, they have a great advantage that you have a very rapid signal because the sampling rates is incredible, so you can resolve many neuronal activity. Slight disadvantage is that you don't know where these neurons are. So they're like somewhere around the probe, and you can build a model. So, and in principle, yes, actually there is one project in the institute when we would like to put the neuropixels into humans during seizures before surgery, but it hasn't been done yet. So potentially, so we have to use the data that is like ECOG recordings or intracranial recordings that people do for just figuring out where the seizure source is. So this can be used, but it's not that precise. So it's always an interplay.
Has any genetic basis for epilepsy been found?
Let me remember. Well, I don't know for sure because I'm not a geneticist, but what I know from other works, for example, there are such a thing as febrile seizures, seizures that have the newborn babies have, and there are very good models, animal models of them. By Kai Kyle in Institute of Helsinki is working on that. But then interesting thing about febrile seizures that they break at some point. So basically, babies have them at some point and then they don't. So because it might be tightly related to the development of the brain. Also, if you look into the total number of seizures, there are a lot of seizures, like in total, in the beginning of life and in the end of life. So it's likely that some environmental factors that interact during the maturating and during aging that might trigger higher chance of seizures. So there might be likely some correlations. Yet I cannot pick up on the top of my head like particular genetic seizure disorder. Yeah.
So it seems like, if I understand it correctly, it's like there are these two cell type, WG1, WG4, that obviously the WG4 seems like the more severe kind of from a morphology point of view. And what your work has been shown is that you can now simulate those neurons correctly, and the simulation, network simulation, also reveal a property that sort of like consistent with what our intuition about the WG1 versus WG4, because the latter seem to be more related to epilepsy, right?
Mm-hmm.
So what will be sort of the next step, like after that? Like what do you plan to do? I mean, first of all, did I understand it correctly? And second,
Yeah. Well, thank you for that brief recap. Yeah, that's very clear. Thank you for summarizing. Yeah, we looked into WG1 versus WG4. And honestly, we have taken the safe route because this is like an easier question in some sense, because people tried to find correlation WG1 versus WG4. But people been working on epilepsy for a while. Our advantage is that we are working at scale, so we can combine different data modalities. We can combine RNA-Seq, we can combine morphology, we can combine electrophysiology, so we can fully characterize this pathology. Regarding the next step, like immediate next step, that these models will be published soon on our website or on GitHub, I'm not sure. But we would like to go to more genetic analysis. So basically, once these models are generated, I will compare the parameter vectors and try to link it to the pathology and try to make it, and hopefully make a prediction for RNA sequence data when it will be available. And also we just did some initial testing using network simulation, so we need to test it in larger networks and more diverse networks with additional cell types. So one of the reasons why we looked into the small networks first, because the larger the network is, the more different combinations of the parameters there are, the more simulations you have to do. So we start with something that works, and then we gradually expand by adding components. Also, we have sampled mostly granule cells. Granule cells that I was talking about come from hippocampus, and basket cells come from cortex, because we didn't have the capacity to sample basket cells. But yeah, we want to scale things up, if it would be possible.
So in terms of the genetic and genomic angle, what might some of the things that we can imagine doing once you understand it? Like, could we imagine even? I mean, this might be completely wacky, but could we even imagine some sort of gene therapy to do epilepsy?
Yeah, I mean, this would be a very exciting potential implication of our finding that might improve the life of patients. So once we identified particular genes that are coding channel, then this would provide us the target, first the pharmacological target that we might try to affect using drugs, or some gene therapy, as you're suggesting, that we would like to specifically modulate these channels a little bit to reduce the propensity for seizures. With respect to that, these comparative studies are very important, where we can look into the VG1 versus VG4, compare between them, look into the differences, and then make a prediction for RNA sequencing data. But this is a long road to go. So, but our reasonable output of this work is that we create these targets by analyzing data, and then it will be the work of a pharmacist or those people who do gene therapy to try it and test it clinically. Maybe some questions from the internet. Thank you.